page 61. Page 61. I jump back and forth between chapter 2 and 3. So we're looking at chapter 3 now, page 61. And these are pretty. These are nice. What were they trying to show you? Just different kinds of cells, right? So would you say cells are all the same way? No. They do different functions, and in the body, if you're doing different functions, you have to probably have different forms. So, do these all have nuclei?
No.
Which one does?
The erythrocyte.
Red blood cells, erythrocytes, do not. They used to. What they do? Well, they spit out their nucleus. Do you think that was smart? No. They're going to die sooner. But they're proud of their work. This will help them do their work. What's their work?
Carrying oxygen.
Carrying oxygen around. And how do you do that? What do you need to do that?
Hemoglobin.
Hemoglobin, that red pigment, hemoglobin. Well, if you had a nucleus in here, it would be taking up a lot of space. But if you spit it out, now you got more room for hemoglobin, so you're able to carry more oxygen. So you're a great worker in the body. It's good, but you're only going to live about 120 days. Life span of a red blood cell is only about 120 days. That's how many months? 4. Okay. So where are worn out red blood cells going? Spleen, the graveyard. Okay. For red blood cells. Fibroblasts are found in connective tissue. And epithelial cells, you know about those. Like epidermis. And yeah, that part of the skin. And coverings in the body have that too. Okay, now I got a question for you. What's this thing? It looks like a loaf of meat or something, sausage or something. What is it?
Skeletal muscle
It's a
cell.
skeletal muscle cell. Are you satisfied? Yes, no? Any criticism?
It's not the technical.
Skeletal muscle cell, I wasn't looking at that so much.
It's got multiple nuclei.
Does it? Here?
Yeah.
They're showing multiple nuclei, so you can't really criticize that. It's not too much bigger than these smooth muscle cells.
Oh, you mean size?
Ah, yes. The size is way too small. It should be spread over the whole page and maybe the next page.
Yeah, but the book says they're not drawn to actual size.
Good. I was wondering why they did this. They purposely did it this way because, where did they say that, at the bottom?
And the top. Well, I mean, for me, it's on the top.
This is
Oh, they're not drawn, they're being careful about it, so we can't criticize them too much. But if you looked at this and believed that this was about the size of a skeletal muscle cell, you're sadly mistaken, because it's much bigger. But as long as you know that, that's okay, all right? So, this is found where?
In adipose tissue?
I'm sorry?
In adipose tissue?
Adipose tissue, right. Because what is it? Yeah, but give me a better name. Adipocyte. Adipocyte, good. Okay. This thing's eating stuff, what's it? Macrophage. So what does it do? Yeah. What?
It eats pathogen.
Phagocytosis?
Yeah, it does eat pathogen. Phagocytosis is what I was after. So that's good. This thing, what would you call it?
A neuron.
What?
A neuron.
A neuron, see? You knew that. What part is this?
That is the
Axon. In the central nervous system, what's a bundle of axons?
Moria? Yeah.
tract.
A tract, good. Would you have known that if we had just gone chapter 1, 2, 3?
No.
No. So you're benefiting from all the stuff we went over before we got to here. So that's why I like to jump from chapter 1 to these other chapters, because you get quite a bit of stuff that now makes sense when you're reading this. You're far ahead in the understanding. Gonads make Gametes, there's a gamete, who is it? Who is it?
Spermatocyte.
It's a spermatocyte or a sperm. Okay, what's the tail called? Flagellum. Singular, flagellum. Okay? So there's quite a bit to talk about there.
The dendrite, the other side.
Dendrites, those little projections on the, neuron cell body.
Gotcha.
What is the
Neurons.
tail again?
This is a neuron cell body, and those are dendrites. These are not. This is the axon with the ends out here. What do we call them? Boutons. They can also be called axon terminals. That's what the book would call it. And your question was? I thought you had a question.
I didn't. Oh, I was asking about the dendrite.
The dendrite? Yeah, I thought somebody asked about the tail.
Oh yeah, I asked what the tail's called for the sperm.
Flagellum.
Flagellum.
Flagellum. Okay, let's go on. Well, we know all cells are certainly not the same. But we could just say, all right, let's make up a model of a cell. And yeah, it doesn't work for all of them, but we threw a lot of stuff together and made the model of the cell here. So what do you see you recognize right away?
Mitochondria.
Mitochondria in their bright red colors. And what about this? Nucleus. What's the fuzz? What?
Chromatin.
Chromatin. And what's the sphere in a sphere? Nucleolus. And what surrounds, what's the boundary on the nucleus? Nuclear envelope. Why isn't it nuclear membrane? It's actually a double membrane. So they call it the envelope. The holes. See the holes? What good are they? Stuff to come in and out. Give me an example of going out. mRNA. The messenger RNA got thrown out of here because it got made in the nucleus because who was there? In the nucleus. Who was in the nucleus?
DNA.
DNA. And you unzipped it by breaking what? Hydrogen bonds. And then you made messenger RNA on it, ripped it off, threw it out there, and then you zipped it back up again. Any questions?
What, zips it together?
The hydrogen bonds come back together.
Oh, they just.
Yeah, they just come back together. I don't think there's an enzyme putting them together, but it might be. Okay, did you notice these are connected? See the connection there? The envelope, the nuclear envelope is connected to the blue stuff with red dots. What's this? Rough endoplasmic reticulum. Why is it rough? Ribosomes who make proteins, right, on here. Okay, but look, you go on over here and we're connected to something that's smooth. Who's this? Smooth endoplasmic reticulum. What does it do? I'll tell you what it doesn't do. It doesn't make proteins. It works on lipids, that's right.
Does it do it similarly, like it gets instructions from the nucleus?
I don't know, I haven't really looked into it, but it does some lipid production and works with it, which is interesting, but I don't fully understand it.
Did you say it doesn't do something?
It doesn't make proteins because it doesn't have ribosomes.
Oh,
So we had the idea that the rough endoplasmic reticulum makes proteins, puts them in water balloons called vesicles, and sends them over to the Golgi apparatus. Which side? The cis side. That's receiving. Then you process it, put a new water balloon around it, and send it out the trans side. Where could it go? Well, it could go over to the cell membrane and then release its contents. What will we call that?
Secretion.
Secretion
There's a blood.
if you're making what? Hormones, and there's a blood supply, like you said, nearby. Yeah, they could go out and go right into the blood supply, and we would say secreting. Like, remember thyroid hormone? What were the two flavors of thyroid hormone? T3 and T4. What was T4? Thyroxine. And how did you make T4? DIT plus DIT, wasn't it? Yeah. T3 was DIT plus MIT. Yeah, good. which was more powerful, T3 or T4? T3. But which did you release mostly? T4. But it converted then to T3 out there on the outside, so it all worked out. You don't have to know that, okay? So let's look at a few other things here. I see a centrosome. What's inside?
Centrioles,
Two centrioles. Look how they're arranged,
perpendicular.
perpendicular to each other. And remember what happens here, microtubules come in and fit in and you make your football shape mitotic spindle. And they're even showing you here, that's a microtubule there. That's what they meant it to be. So they showed some microtubules. Now what else do we see here? We can see some more stuff. We can see a lysosome. Lysosome is full of what? Digestive enzymes, strong digestive enzymes. What are you going to use them to do? Well, if you've got leftover pieces of cell organelles, or if you have dead material or whatever, lysosomes can break it down and then it can be recycled into something else. Strong digestive enzyme. What would happen if all the lysosomes broke open?
Apoptosis.
Yeah, probably. I don't know if they call it apoptosis, but the cell would be goner. That'd be it. Okay, so it's kind of not a good idea. Okay. I'll tell you that later. Okay, I was thinking about telling you something about it. So what is this? It's a mitochondria with how many membranes? Two outside smooth, inner one folded. The folds are cristae. And what's on the cristae? Enzymes to do what?
Make ATP.
Regenerate ATPs. Where are you going to get the energy? Glucose, food. And you're going to take that energy and regenerate ATPs. That's good. That's a nice, worthy cause. All right, well, that was good. Oh, I see somebody else here. Look at, don't I? Maybe I don't. I thought I did. Yeah, right down here. Looks like a lysosome. You really wouldn't know the difference just by looking. This is a peroxisome. 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 It has two types of enzymes inside. Two types. Peroxisomes are supposed to help you. They're really good. Because, have you ever heard of free radicals? Anybody take vitamin C for the free radical treatment? In other words, you're trying to get rid of free radicals. Vitamin C is one of the possibilities. I'm not thinking what the name of it is that we talked about.
Ascorbic acid.
Ascorbic acid, yeah, but there's a name for the things that do that. Free radicals.
Antioxidant.
Antioxidants, that's it. Antioxidant to repair damage. Free radicals damage you. And you say, well, I just won't eat them. I'm sorry, but your metabolism naturally makes free radicals. So you have to do something about it. And this will, peroxisomes help, but antioxidants are supposed to help that too. And people do take antioxidants with the idea of trying to counter free radicals and the damage they actually do to a cell. So peroxisome has two groups of enzymes to deal with those free radicals. So the first group that works on the free radicals are peroxisomes. No, peroxisomes, the thing here, oxidases. So first, free radicals get treated with oxidases, and then that makes hydrogen peroxide. What do you think about hydrogen peroxide? What do you use hydrogen peroxide for? If you've got cuts or wounds, you can pour hydrogen peroxide on it, bubbles up. Okay, it's helpful. But in your body, it's not supposed to be so helpful, but some people do put it in their bodies intentionally. But hydrogen peroxide, question?
Ingesting?
IV. IV.
What does that do? Like, you don't.
Well, they think it gets rid of some bad stuff.
They're not exactly.
But it's not so good.
I can't
Okay.
imagine like putting hydrogen peroxide
It may be a low concentration or something like that.
Oh,
They do it. Well, here, the peroxisome treats the free radical and makes hydrogen peroxide. Then, that's from the oxidases. And then the hydrogen peroxide gets treated by catalases. And they change now this hydrogen peroxide into water. So you went from free radical to water. I would say that was a success. The water's not gonna hurt you. Isn't that neat? Take something that's really bad, change it to water. 2 steps. I mean, you had to do the oxidases and then the catalases. How do I remember which is which? Well, I can tell you this, because that's what I use, but you can do whatever you want. I think about the cattle being down at the water. So catalases make the water. That's the
And
memory
that's-
device.
that's enzymes? Like
Yeah, those are
different
enzymes, the catalases,
Peroxidase.
aces, yeah, oxidases and catalases. But they're in the peroxisome, so you can treat free radicals with peroxisomes. So there's interesting stuff in the cell here. We wanna keep on going. How many strands of chromatin? 46. Let's go on. I like these 2 pages because it's very nicely done. This is 64, 65. What's the first thing you do when you look at a picture? Get oriented. So what are we looking at, Blessing? What are we looking at? Somebody thinks said it. What? Cell membrane. It's made of phospholipid bilayer. It's composed of a phospholipid bilayer. Yeah, cell membrane. Okay. So what do you know about phospholipids?
They have like a hydrophobic end.
They have a part that likes water and a part that hates water. Which part likes water?